Hello, everyone. Welcome to the third and final part of our lecture on the heart. So previously we talked about the overall structure of the heart, the four chambers, the fact that we had two circulatory systems, the pulmonary and systemic. So now that we've covered the basic structure of the heart, let's see how the heart functions. So if you remember, heart is made up of cardiac muscle. So cardiac muscle, one of the ways we're able to identify that in our tissue laboratory is these things called intercalated discs or intercalated discs. So what these intercalated discs actually are masses of desmosomes and groups of gap junctions. So if you remember, a desmosome was like a rivet. It really kept two cells tightly connected to each other. And that's because, right, the heart is going to undergo a lot of strain, a lot of stress, generating all that pressure. So it needs something to hold the cells together, hold the cells together. And those are going to be the desmosomes. Now, gap junctions were little tubes that connected the cytoplasm of one cell to another. And that's going to be very important for propagating the signal for a cardiac cell to contract. Remember, cardiac cells are short. So one cell will tell the neighboring cells it's time to contract. This is fundamentally different than what we saw with skeletal muscle. Skeletal muscle, each long skeletal muscle cell had a separate attachment to a neuron. It's not the case here in the heart. Okay, so here is our Y-shaped, more or less Y-shaped cardiac muscle cell. It has one or two centrally located nuclei. They're not pushed off to the side. We still have myofibrils that still have sarcomeres and all the structures, at least as far as the sarcomere is concerned, we also see here as in skeletal muscle. If we take a blow up of those intercalated discs, right, that's just the space, the union where two cells butt up against each other. You can see lots of desmosomes to hold the two cells together and lots of gap junctions, which are going to serve as electrical synapses. So one cell is going to share chemical signal, an electrical signal to contract with the neighboring cell, and it will pass through those gap junctions. So this is the basic structure of skeletal muscle cell. We kind of already covered this, but it's good to review it. We're still going to have triads. We're still going to have sarcomeres. We're still going to have the transverse tubule and the terminal cisternae. So very, very similar in the specific cellular structure that we saw for skeletal muscle. If we look at an image of the histology, you can see those intercalated discs made up of gap junctions and of desmosomes. So this is pretty much review what we saw in skeletal muscle. Now, let's talk about how these cells are able to receive. How do you start generating a heartbeat? And the interesting thing is, heart cells tell themselves when to beat. Your brain does not originate a signal for the heart to start contraction. Your brain can moderate your heartbeat. It can speed it up or slow it down, but it doesn't actually start the beat of the heart. The heart tells itself to beat. And this ability is referred to as autorhythmicity. So heart is capable of initiating its own contraction. And there are specialized groups of cells that are responsible for that. And they're referred to the pacemaker cells, or pacemaker of the heart. So the pacemaker is located in the SA node, Cyanoatrial Node. This is located on the posterior wall of the right atrium. These cells will spontaneously depolarize and start passing action potentials from one skeletal muscle cell to another, and then it will expand across all the atria, and then all those cells will start contracting one after another, just like a row of dominoes. So the impulse that triggers the cardiac cells to contract starts at the AV node, starts at the SA node, going back here. The signal to contract starts at the cyanoatrial node, the SA node, and is going to spread out over both atria. Now, as it expands from the top of the right auricular wall, right, those potentials will then hit a second area called the AV node, the atrioventricular node. And what this is going to do, it's going to receive the signal that was started back at the SA node, and it is going to transfer that signal to a structure called the AV bundle. The old term for this is bundle of His. And the bundle of His is going to run in that interventricular septum, and it will split into two bundles, a right bundle and a left bundle. Those bundles will then split into structures called Purkinje fibers, and the Purkinje fibers are going to continue that action potential in the ventricular wall. So order of progression, SA node through the wall of the atrium, through the auricular wall, to the AV node, from the AV node to the bundle of His, to the right and left bundles, to the Purkinje fibers, back to the heart cells. Let's take a look at an image to make this a little bit more clear. So here we have the cyanoatrial node in the posterior wall of the right atrium. These are the cells that are capable of generating an action potential. That action potential travels through the wall of the atrium, travels through the wall of the auricle, both from the right auricle, superior to inferior, and from the right auricle to the left auricle. So that's what this image is shown here. The green is the image to contract, starting at the SA node, traveling inferior to the AV node. The AV node then picks up that signal and transfers it to the bundle of His, shown here. Right? The bundle of His then separates into the right and left bundles. Right and left bundles travel down to the apex and then come back up, and then they split into Purkinje fibers, and the Purkinje fibers then reconnect to the cardiac muscle cells, and they start contracting. This is why the heart beats from the top down and bottom up. Top down, bottom up, because superior portion of the auricle starts contracting top down. If you think about it, this makes sense, because that allows the auricle to inject blood into the ventricle. The ventricle doesn't have a hole in the bottom, it has a hole at the top, right? So the in hole and the out hole are both at the top of the ventricle. So the atria squeezes top down to inject blood into the ventricle. The ventricle squeezes from the bottom up, and that way blood is ejected out the roof of the ventricle. Now, you may be thinking to yourself, what's going on here, right? If the signal to contract can simply be propagated from cell to cell to cell, why doesn't the heart simply beat from the top down and blow a big hole out the bottom of the heart? Because if these cells are generating the action potential, the signal to contract, it should just travel from cell to 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 the bottom of the heart, to the apex of the heart, and the whole thing just contracts like that. Well, do you remember back in lecture 2, I said something about the fibrous skeleton? Where is that guy? Here, the fibrous skeleton. I told you it was a structure that's going to physically support the 4 valves of the heart and serves as an electrical insulator. That was the key point. So the fibrous skeleton prevents that signal started in the SA node from directly reaching the ventricle. Let's go back to our conduction picture. So here, the signal to contract is started at the SA node, and it's going to travel throughout both atria until, boom, it's blocked. As soon as it hits that fibrous skeleton, which is laying right here, it's stopped. It cannot proceed because the fibrous skeleton is acting as an electrical insulator. But we still need to get the ventricles to contract. So how do we get that signal? Should we have a completely different way of generating a heartbeat down there and then coordinating it with the SA node? That's too complicated. So we have this short circuit, or we have this bridge, you can think of it. The bridge is the bundle of His. So the bundle of His will take that signal, once it reaches the bottom of the atria, and transfer it to the bottom of the ventricles. So going through this step by step, we generate the signal to contract at the cyanoatrial node in the superior posterior wall of the right atrium. That signal will go through the gap junctions, through the intercalated discs, cell to cell to cell, until it hits the AV node. The AV node then picks up that signal, transfers it to the bundle of His. From the bundle of His, it splits into the right and left bundle. Right and left bundle connect to the Purkinje fibers, and the Purkinje fibers then transfer that back to the cardiac cells. And now the heart muscle, the myocardium of the ventricles, starts to contract. So SA to AV, that's what's shown here. Then it's going to go down the bundle of His to the right-left bundles to the Purkinje fibers, and back to the myocardium of the ventricles, back to the cardiac cells that are located in the ventricles. So the heart is going to beat from the top down, bottom up. That way blood goes through the atria into the ventricle, and when the ventricle contracts, it ejects blood out the ventricle into the arteries of the heart, either the pulmonary trunk or the aorta, depending on which side of the heart you're talking about. Okay, so the heart is going to be innervated by both divisions of the autonomic nervous system. Remember, the autonomic nervous system is going to control unconscious motor activity. So typically, if you're going to get the sympathetic nervous system activating, it might slow the heart down. If you're going to have, parasympathetic is going to slow the heart down, and sympathetic will speed things up. Now, there's simply a big mess of autonomic motor neurons around the heart, and we're just going to call that the cardiac plexus. Remember, plexus is just interweaving neurons. Interweaving axons, interweaving nerves in this case. So what's the big takeaway here? The autonomic nervous system does not generate the heartbeat, but it can help control the activity, speed it up, slow it down, increase cardiac output, decrease cardiac output, and that's just how much blood is actually ejected for each squeeze, controlled by the autonomic nervous system. Okay, now let's get into the actual functioning, the steps of a heartbeat, and it's called cardiac cycle. So because this is anatomy, we have to have fancy words. So when the heart contracts, we're going to call that systole. So when the heart squeezes, that is going to be called systole. When the heart relaxes, that's going to be called diastole. Right? Perhaps you've heard of systolic blood pressure and diastolic blood pressure. Systolic and diastolic. Systolic is when the heart is contracting. Diastolic is when the heart is relaxing. Yes. Okay. So what is happening in ventricular systole? Now, remember, the walls of the auricle are very, very thin, right? You only need enough strength to send blood from the atrium into the ventricle. So when you're talking about moving blood in the body, moving blood from the heart to the lungs, moving blood from the heart to the rest of the body, you're talking about the action of the ventricles. So here we're just looking at big picture. What's the big mover? Ventricular systole. So in ventricular systole, you're having full contraction of the ventricular myocardium, just the wall of the ventricle. These muscles are all getting tight, they're all squeezing. That is increasing pressure in the ventricle. Increased pressure in the ventricle causes the AV valves to close, and it will cause the semilunar valves to open, right? So blood can get out. So that is what we're seeing here. Ventricular systole, AV valves close to prevent blood from flowing backwards into the atria, and semilunar valves open. Because the heart is contracting, blood is being ejected into the arteries. So this is what ventricular systole looks like. Contraction of the ventricles means AV valves close, semilunar valves open, blood is ejected from the heart. Now, when we look at ventricular diastole, the opposite is going to be the case. The muscles have completely contracted, now they're relaxing. When they're relaxing, the AV valves open because now you're going to have more pressure in the atria than you do in the ventricles, and that pushes down on the parachutes, so blood is able to flow in from the atria through the AV valves into the ventricles. The one-way valves are pointed the opposite direction for semilunar. Here, at the semilunar valves, the pressure in the arteries is greater than the pressure in the ventricles, but the one-way valve is pointed the other direction. So when blood presses from the arteries to try to get back in the ventricle, the valve slams shut and is prevented from doing so. So this is a picture of what ventricular diastole looks like. AV valves open, semilunar valves close. Now, you definitely want to have these things very clear in your brain. Ventricular systole. In ventricular systole, ventricles contract, AV valves close, blood is ejected from the ventricles. In ventricular diastole, the ventricles relax, AV valves open, and semilunar valves close. Now we have a name for this, right? This is lub, this is dub. So these are the heart sounds, lub dub, right? Pitter pat, lub dub. This is the alternating sound of blood first slamming into the AV valves, causing them to close, and dub is blood trying to get back into the heart and slamming into the semilunar valves and them closing. So this is lub, this is dub. Lub, ventricular systole, causing AV valves to close, blood hits those valves, makes the lub sound. In dub, ventricular diastole, blood tries to backflow into the ventricles, causing the semilunar valves to close. That makes the sound dub. So lub, dub, lub, dub, lub, dub. Do you need to know what's lub and what's dub? Who's contracting, who's relaxing, who's open, who's closed? Absolutely, absolutely. Okay, this is a little bit complicated, so I'm going to come back to it, but I'm going to do this figure first. So this thing here is super important. Every little bit of information on here, I'm going to want you to memorize. So put a bunch of stars across the top. So what this is showing you is the flow of blood through the heart, through both circuits. Notice that up here, we're going to have blue. Blue means deoxygenated. If it's deoxygenated, this must be the right side of the heart, because the job of the right side of the heart is to take deoxygenated blood and send it to the lungs. Now down here, this is all red. If it is red, right, the mover here is going to be the left side of the heart, because the job of the left side of the heart is to take that freshly oxygenated blood from the lungs and send it to the rest of the body. Where you have a color change, blue to red or red to blue, means you have a capillary bed. So if you're going from blue blood to red blood, you have to be giving up carbon dioxide and bonding oxygen. Where does that happen? That happens in the lungs. That's why we have this purple box here. This is the capillary bed in the lungs. This is your pulmonary capillary bed, which takes deoxygenated blood and oxygenates it. Likewise, on the other side of the body, we're going to have systemic capillary beds where the opposite takes place. Oxygen will be released, carbon dioxide will be bound. So this little box here represents systemic capillaries, where you go from red blood to blue blood, and the whole thing just goes around in a big circle. So let's walk through the various structures of the heart. We're going to start in the systemic veins. Remember, what's a vein? A vein is a vessel that takes blood towards the heart. We have two kinds of veins. We have pulmonary veins and we have systemic veins. We're starting in the systemic veins. So this is going to be deoxygenated blood that's lost its energy. So blue ooze, deoxygenated, low pressure. This is going to drain through all the veins of the body, eventually joining to form the superior and inferior vena cava, shown here. Blood then flows from the superior and inferior vena cava into the right atrium. Right atria starts to contract. That's going to send blood through the right AV valve, through the tricuspid. Send blood through the tricuspid into the right ventricle, shown here by this triangle. The right ventricle is going to contract, and that generates enough energy to eject blood out the pulmonary semilunar valve into the pulmonary trunk. The pulmonary trunk, this is an artery. This is blue squirt. Why is it blue? Because we have not yet been to the lungs. So it remains blue. Why is it squirting? Because we're leaving a ventricle. If you're leaving a ventricle, you're under high pressure. That means you squirt. Arteries always squirt. Veins always ooze. But that rule does not apply to color. Sometimes arteries are red if they're systemic, but here we have an example of a blue artery if it's part of the pulmonary circuit. However, go back to our definition, an artery always carries blood away from the heart, which means it's always connected to a ventricle. A ventricle is where you generate pressure, so arteries always squirt. Veins carry blood back to the heart. Veins always connect to atria. There you have low pressure, so a vein or an atria will always ooze. Let's start over again. Systemic veins drain blood from the body, eventually to the superior and inferior vena cava. That drains into the right atrium. Right atria contracts, sends blood through the tricuspid valve into the right ventricle. Right ventricle contracts, sends blood out the pulmonary semilunar valve. That goes to the pulmonary trunk. Pulmonary trunk splits into the right and left pulmonary arteries. That takes blood to the lungs. Now, all that high pressure blood going through the spongy material of the lungs, it's going to lose that energy. It's going to lose that pressure. It's also going to bind oxygen and give up carbon dioxide. So we're going to get a pressure change and we're going to get a color change. So on the other side of the lungs, we're going to have red ooze. Red because the blood has gained oxygen, and ooze because it's lost its pressure. Now, because it's lost its pressure, it needs to go back to the heart before it can go to the rest of the body. So if we are red blood headed back to the heart, that means this is going to be a vein, because veins carry blood back to the heart. This happens to be a red vein. So this is a pulmonary vein, draining the lungs back to the heart. Pulmonary veins lead to the left atrium contracts, sends blood through the bicuspid or the mitral valve into the left ventricle. Left ventricle contracts, that's the big squeeze. That's the big, thick wall of the heart, right? That left myocardium, big squeeze. That's gonna force blood out the aortic semilunar valve and into the aorta, and from the aorta into the systemic arteries of the body. And that sends the oxygenated blood at high pressure throughout the body, where it goes to those hungry cells, those hungry tissues, where you're going to have a capillary bed where oxygen is released, carbon dioxide is bound, and now you're going to have another change of color and another change of pressure. So red high pressure blood, red squirt. Once it goes through another capillary bed, another sponge, it loses its pressure, so it's going to turn to ooze. It's lost its oxygen, so it's going to turn blue. So systemic veins, blue ooze. So go through this cycle multiple times. You want to have this super, super clear in your head. Each of the various structures and what order you go through them. Know, is it red? Is it blue? Does it ooze? Does it squirt? And why? Right? This is really important to understand the parts of the heart and the functioning of the heart. So make sure, again, a whole bunch of stars across the top here. I tend to ask lots of questions on this. When we get into vessels, I'll start pinning vessels and say red ooze, blue squirt, blue ooze, red squirt, you know, all those kinds of stuff. So definitely have this like super clear in your head. Now, to go into a little bit more detail, we're going to go through this figure here. So in this figure, we have a slightly more complex version of the cardiac cycle. So it will start with atrial systole. Okay, atria is a small superior space, systole is contraction. Okay, so this is contraction of the atria. Both atria contract, sends blood through the AV valves into the ventricles because the ventricles are relaxed. When the atria contract, they have just enough pressure to overcome and open the AV valves, and blood will flow into the ventricles. Next step, early ventricular systole. So the ventricles, the myocardium, just starts to contract. Not fully, it's just starting. What this does is this is going to raise the pressure inside the ventricles just enough to close the AV valves, right? To close the tricuspid and to close the bicuspid valve. Dub. Excuse me, lub. This is lub. Closing of the AV valves is lub. However, in early ventricular systole, we have not yet generated enough pressure to overcome the pressure in the arteries. So the semilunar valves remain closed. So we have an increase in pressure to close the AV valves, lub, but we have not yet been we have not yet generated enough pressure to overcome what's in the arteries, so semilunar valves stay closed. Then in late ventricular systole, we have full compression of the heart wall. We generate maximum pressure, AV valves stay closed, but now the pressure inside the ventricle exceeds the pressure in the arteries. AV valves, excuse me, semilunar valves open and blood is ejected from the heart. And the next step, early ventricular diastole. So the ventricles have completely contracted. Now they're starting to relax. As they start to relax, not fully relax, but when they start to relax, the pressure inside the ventricle drops lower than the pressure in the arteries. Because you now have a higher pressure in the arteries, the semilunar valves slam shut. Dub. So when the semilunar valves slam shut, this is dub. However, the pressure inside the ventricle has not yet dropped below the pressure in the atria, so the AV valves now remain or continue to remain closed. Only in late ventricular diastole, only when the ventricles have completely relaxed, will the pressure drop low enough for blood to start pouring, start flowing in, mostly through gravity, pouring in from the atria into the ventricle. And now we can repeat the entire process again. So I'll go through this one more time and hopefully I won't trip over my tongue so many times. So we will start with atrial systole. The atria, auricular walls contract. This generates enough pressure to drive blood from the atria through the AV valves into the ventricle. Then in early ventricular systole, the ventricles just start to contract. That generates enough pressure, so the pressure inside the ventricle is greater than the pressure in the atria, so the AV valves slam shut. However, the pressure is not high enough to overcome the pressure in the arteries, so the semilunar valves remain closed. So in this step, both valves are closed. When the AV valves close, that's lub. Moving on to late ventricular systole, complete contraction of the ventricles. Now we have maximum pressure. Now the pressure in the ventricles is above, it exceeds the pressure in the arteries, and then semilunar valves open and blood is allowed to escape. Then the ventricles just start to relax, not fully, they just start to relax. The pressure inside the ventricle drops below the pressure inside the arteries. That causes semilunar valves to close, right? Semilunar valves to close. Dub, lub, dub, lub, dub. This is lub, this is dub. In this step, all four valves are closed. In this step, all four valves are closed. Then finally, in late ventricular diastole, we have complete relaxation of the ventricular wall. Now the pressure in the ventricle drops below the pressure in the atria and the AV valves open. Semilunar valves remain closed and we're ready to start the whole process over again. So this is also another very important figure, right? Make sure you can follow the steps moving around and around and around and know why certain things close at a certain time. It has to do with the functioning of these valves, four valves, right? They're not little motorized gates that open and close. They're just controlled by the pressure difference between the atria and the ventricle. So with that, that concludes our section on the heart. My suggestion, other than simply just learning the structures of the heart, spend a lot of time in this figure here. Be able to recite this blind, right? From memory, be able to walk through each and every single one of these steps, right? Once you have that, then move on to this figure and understand the timing of the change in valves. And the key to understand that is to remember when the pressure inside the ventricle exceeds the atria, AV valves close. When the pressure inside the ventricle exceeds the pressure inside the arteries, semilunar valves open, right? And then down here is reverse. If the pressure is higher in the arteries than it is in the ventricles, the semilunar valve closes. If the pressure is lower in the ventricles than it is in the atria, then the AV valves open. So go ahead and draw it out. Make sure this is super clear in your head, because it's really important to understand what exactly is going on, right? How does the histology, the structure of a cardiac muscle cell, require the need for an SA node, AV node, bundle of His, Purkinje fibers, and how it relates to the heart cycle? Because this is going to give you the basic knowledge to understand things like how exactly an EKG works, right? What do those little bumps mean? But you understand something about cardiac tissue, understand about the electrical wiring of the heart, how the various chambers of the heart are coordinated. You can start to understand exactly how an EKG works. And with that, I'll wrap up the heart and see you guys next time. Take care.